Okay, so it's my real pleasure to introduce our this afternoon world's speaker, George Ross. George is a Krieger Eisenhower professor at Jansky's Department of Biophysics, Johns Hopkins University. George has had an enormous impact on the field of protein structure and folding. In fact, I hope it will be okay to say that George turned 65 not so long ago, and there was a whole issue of the Journal of Protein that was devoted to the impact that his work has on the protein structure and protein folding field. George is recognized for his contribution to understanding of the hierarchical organization of protein structures and the relation of that organization to protein folding, which he also proposed to be hierarchical in nature. He studied in depth secondary structure and the forces that stabilize those structures. In particular, in 1988, he published a prominent Science article which provided the initial description of the specific termination signal at helices. And that article has led to an enormous body of both experimental and computational work on understanding of formation and stabilization of helical structures. Among more recent work related to secondary structure is George's work on stabilization of secondary structure through solvent and the solvent accessibility. Again, going back in time, in 1980, at the time when the protein structure field considered everything that is not a helix or strand to be a random coil, George demonstrated that many so-called unordered proteins, in fact, are quite ordered and can be considered secondary structures themselves. In particular, he introduced so-called omega loops. More recently, George has turned his attention to the organization of, again, presumably random coils that makes unfolded proteins. He reevaluated the assumption that unfolded proteins can really populate extremely large sequence space, structure space, and show that really this random coil is, again, not as random as one might think. George also made enormous impact on the current understanding of the role of hydrogen bonding and packing in determining protein structure. Finally, to those like myself who were fortunate to work with George, I think one thing that we can observe is his striking feature of George's approach to science is the following. He often looks at the things that are obvious to everybody and then say, actually the things are not so obvious as they appear to be. But at the same time, things that, are obvious, his points are quite often missed, and only after they're being noticed are being accepted as being obvious. And I guess his today talks, which title is Protein Folding, Seeing is Deceiving, will be a testament of this views.
Thank you, Teresa. Thank you for the kind and detailed introduction. And thank all of you for waiting here through all of our startup problems. What we've ended up doing is to take my talk, which was on one computer, and transfer it to another computer. So it's almost guaranteed not to quite work. I hope you'll bear with me. And let's begin. So what I want to do today is to lead you through a fairly long story, and I'm just going to hit some high points for this long story because I want to take you through the long narrative. And the narrative has to do with protein folding, of course. And what I'm going to propose is that the model that we've been using these many years is probably been misleading us. It's good enough to be able to incorporate current experimental information into the model, but nonetheless, I think the model has probably been misleading us in many respects, and I'm going to suggest an alternative model. So just a couple preliminaries first. What do we mean by protein folding? There are many definitions of protein folding that are floating around these days, but the definition that I will use for this talk is the classic definition, which is how do you predict the sequence of a protein? How do you predict the structure of a protein if what you knew was its sequence? And here's the sequence of ribonuclease A, 124 amino acid residues, and under physiological conditions, what it does is that, and the question is how. And if you really understood how, could you take that sequence alone and predict that structure? We know it should be possible in principle because Anfinsen showed us that that's so. Who cares anyway? Well, I claim that we all care. Understanding how this works at the deepest level will have a profound impact on our understanding of protein-based life. And of course, it has many practical implications as well. And rather than express this in my own words, here's a quote from Sidney Brenner that appeared in Science not all that long ago. And I love this quote. In my own subjects, genetics and molecular biology, research has become so directed toward medical problems and the needs of the pharmaceutical companies that most people do not recognize that the most challenging intellectual problem of all time, the reconstruction of our biological past, can now be handled with some hope of success. The problem itself was first articulated, we used to teach, by Mirsky and Pauling in 1936. John Edsel showed us that in fact Hsien Wu had published much the same idea as Mirsky and Pauling in 1931, but it was published in the Chinese Journal of Physiology that most of us don't read. And Edsel republished it much, much later in Advances in Protein Chemistry. Many of the current ideas are already here in the Mirsky and Pauling article. But in fact, the way we tend to think about this now is the way that Chris Anfinsen taught us to think about it during his work here at the NIH in the 1950s. It's the spontaneous acquisition of the native conformation under physiological conditions, which means that when you take a protein and subject it to physiological conditions, aqueous solvent, body temperature, a little bit of salt, that spontaneously, without burning any ATP at all, it folds into its native-like structure, its biologically relevant structure. So it is a disorder to order transition, and we want to understand how this works. The problem at this deepest level remains unsolved after 3/4 of a century. It is fundamentally a thermodynamic problem. Thermodynamics is arguably our most powerful tool for understanding biological systems. So why is it taking us so long? And I'm going to suggest that one reason it's taking us so long is because we've been using at least some of the wrong premises here. Which is not to say that there hasn't been progress. There's been substantial progress in recent years. Most of the progress has been based on one form or another of pattern recognition. You take fragments from proteins of known structure, you put them together in a library, you have a large library of many fragments, and you find a way of stitching them together in a self-consistent way and evaluating their energy in one way or another. And that kind of pattern recognition approach has been a major success in recent years, even more successful when coupled with some experimental information like chemical shifts. So these are all very welcome advances. But fundamental physical-chemical understanding still remains a deep challenge. And so we'll get back to this in a little bit. So here's what the talk today is going to be about. Here's the preview. First, we'll go through what we know. And what we know and really think we know, in fact, comes from work that was done at the NIH. The Anfinsen Hypothesis and the two-state model from Ginsberg and Carroll, and we'll talk about that briefly. And so what have we come to assume, either from what we know or from what we've subsequently learned? And here are the kind of, if you could wake up a textbook writer like Jeremy Berg in 2 a.m. and say, Jeremy, tell me the way that protein folders think about protein folding. If he wasn't too grumpy at that hour, here's what he'd say. He'd say, first of all, the native state is the lowest energy conformation. That's a consequence of Anfinsen's work. The unfolded state really doesn't have much in the way of organization. We describe it statistically, like a random coil. Since all backbones are essentially the same, you could argue that glycine and proline might be exceptions, but essentially all backbones are the same, and so the fold has to be selected by interactions between and among the side chains. The predominant organizing interactions are hydrophobic. It's the hydrophobic effect that folds proteins, and those organizing interactions remain visible in the folded structure. So you can look at the folded structure, see who's contacting whom, and that will give you a very strong clue about how those interactions deployed early in the folding process and persisted through the folding process and stabilized the native state. Those are that's a sort of caricature of the way people have thought about this. Those are the underlying assumptions. We're going to question all of these assumptions in order and propose an inverted model. So this is a bold proposal. Turn your skeptistats all the way to max, and here we go. Oh, and one other thing. I'm going to cover a lot of material here, so if you fall asleep in the middle of this and then wake up before it's over and wonder where we are, I will have down here in the right-hand corner a little icon which will tell you where we are in this talk in terms of going through. So feel free to fall asleep and tune back in by looking at the lower right corner. So here are the anchoring ideas. See, it says what we know. Here are the anchoring ideas. Here's Anfinsen's work. What he showed in the case of ribonuclease is that under unfolding conditions, high temperature, denaturant, you have a diverse population of unfolded proteins, but under native conditions, the population goes predominantly to a single folded structure, which is represented here as a cartoon. But if you look in the protein database at this point, there are approximately 67,000 structures. When I looked a couple days ago, of course, it's not 67,000 unique proteins. Many of them aren't proteins at all, and there are many variants of proteins. But in any case, there are about 50,000 proteins of known structure sitting in the PDB. So we know a lot about this state, and this state we tend to describe statistically. Anfinsen showed that ribonuclease folds spontaneously to this state under physiological conditions. How is he thinking about it? Here's Chris Anfinsen. He spent the last 10 years of his life or so at Hopkins in our biology department, but this is his famous work that was done here at the NIH. And so here is the way that people tend to think about this, Chris included. In fact, I spoke to him, and it is the way he thought about it. What you have is the folding reaction shown here as a thermodynamic equation. What's really happening? Well, under unfolded conditions, you have a population of proteins. I don't know how many. The number of proteins you might have in some experiment, say 10 to the 15th protein molecules. And they're in solution, and you subject them to unfolding conditions by putting in denaturant or raising the temperature. Now, under unfolding conditions, each of these molecules is free to adopt a given conformation at any time slice, but the energy differences between one conformation and another conformation are very small under these conditions, of order physiological KT. And so any given protein molecule is free to wend its way from one conformation to another conformation because it's always getting these little KT kicks in the pants. So if you look at your population of 10 to the 15th molecules, they're distributed over this very broad landscape. Now you switch to folding conditions, and under folding conditions, the entire population wends its way to the folded state. Presumably what's happening is that under folding conditions, side chains are beginning to interact with one another. Some of the interactions are favorable, some of the interactions are unfavorable, some of the interactions are extremely favorable. Presumably then, the constellation of interactions between and among side chains that corresponds to the native state of the protein is so much more favorable than any other distribution of these side chains that it selects the native state from all other possible conformations. And it does that, the energy difference between that conformation and the next conformation is sufficiently large that what you see is that that particular conformation being populated in 99.999% of the population. That's the way that people tend to think about it, Anfinsen included. But of course, it really doesn't make any difference what our model is. This is thermodynamics. That's what it does, and we know that that's what it does, and we can speculate about how it does it, but thermodynamically speaking, we know that you populate the native conformation in essentially 99+% of the total population under folding conditions. Here's the next thing we know, or think we know, and this also comes from work done at the NIH by Ginsberg and Carroll. It's another old paper, 1965, and what it shows is the fraction of unfolded molecules, this happens to be ribonuclease again, as a function of some denaturing property, in this case temperature, but it might well have been denaturant concentration. And what you're seeing here are a number of different kinds of tick marks. Each of these kinds of tick marks represents a different probe of the native conformation. Almost all of them are spectroscopic probes. One of them happens to be intrinsic viscosity. They're very different in kind. And yet, after suitable normalization, all these probes fall on the same unfolding curve. So what that suggests is that this is a highly cooperative process. To a physicist, it would look like a first-order phase transition. And what you're seeing is one of these sort of all or none kind of changes, so that if you would go to the midpoint of this curve, instead of seeing the molecules halfway unfolded, what you would see is half the molecules are still fully folded and half the molecules are still unfolded, or completely unfolded, and the population of intermediates is absolutely negligible. So that gives rise to this idea that many small proteins fold in this kind of two-state manner. They're either folded or unfolded with a negligible population of intermediates. So the current paradigm that comes from all of that is that you go from the random coil, the unfolded population distributed over this broad energy landscape, to the folded population. And the only way that you can explain that is that the native conformation is very low in energy compared to all the others. So the native state corresponds to a minimum energy conformer. Now we know, and Anfinsen was very careful to articulate, that that means minimum in Gibbs free energy, which includes the free energy of the entire system, including the solvent. But people have taken a shortcut in their thinking, which goes something like this. I'll caricature it again. When the protein folds, it folds to a single conformation. We know it's rattling around in wells a little bit. We know that it doesn't stay 100% folded, that it's fluctuating, it unfolds, there's partial unfolding, there's complete unfolding, but at any given instantaneous moment in time, 99.999% of the molecules are folded for a typical globular protein. And so what we argue is, well, basically it forgets it had any entropy to lose. And so what it must be doing is minimizing its energy. The entropic component of this is largely gone in the folded conformation. And it remains in the folded conformation, so clearly that's a stable conformation. Furthermore, the unfolded state is a featureless landscape. People have explored this. We'll get back to that in a bit. Since the unfolded state is a featureless landscape and there are no stabilizing interactions there, the folding must arise on the n side of this U to n equation. All backbones are essentially the same, so it's really the side chains that have to discriminate the correct fold from all other folds. The predominant organizing interactions are hydrophobic. It's the hydrophobic effect that folds, and these organizing interactions remain visible in n. And so it's given rise to this current metaphor that under the unfolding conditions, the protein can explore this broad energy landscape, but it's rather undifferentiated. And then as it loses conformational entropy, it falls down to this steep minimum where favorable enthalpic interactions kick in, but entropy, which is represented by the width of this, is lost. That's the paradigm. And now we're going to question it. So, we move on to the first question, really whether the native state corresponds to a minimum in energy. So here is this Ginsburg and Carroll experiment represented diagrammatically. Here what you're doing is denaturing the protein, and the fraction of unfolded molecules goes up as the denaturant concentration goes up. And if this is two-state, as we tend to think for these small molecules, and which can be proved rather rigorously for a large number of molecules, then what that says is that the free energy difference between the folded and the unfolded state is given by minus RT times the log of the concentrations of the native population divided by the unfolded population. And at this midpoint, what we know is that the free energy difference between the native and unfolded has to be 0. When these concentrations are equal, it's log of 1, and so the free energy difference has to be 0. So if we begin to do things to destabilize the protein, which often we do even though we didn't want to, we put in a mutation with the intent of making more stable proteins. Typically what they do is make a less stable protein. But nevertheless, what happens is that, so we reduce the free energy difference between the native and the unfolded state, but have we wrecked the protein when we've done that? And the answer is no. We've moved up on this line. And as we continue to move up on this line, if we go to the midpoint where the free energy difference between the native and the unfolded state is now 0, still it says that half the population knows how to fold into the native state. In fact, if we destabilize further by going way up in this curve, where we probably can't experimentally access that population, still what it says is that under those conditions, most of the population is unfolded, but a remaining small subpopulation remains folded. And if it remains folded in its native state, that means the information necessary to encode the native fold is still there, even under strongly destabilizing conditions. DNA biochemists know this in their gut. The reason, if I tell you the sequence of the Watson strand of DNA, you can tell me the sequence of the Crick strand because of hydrogen-bonded complementarity. But the reason why DNA folds as double-stranded plectidemic DNA, what stabilizes it in that form, has much more to do with base stacking than it does with hydrogen-bonded complementarity. So DNA biochemists make this distinction between what stabilizes the DNA and those specific interactions which tell you what the Watson strand is if you know what the Crick strand is. Protein biochemists tend to conflate these two and say, well, it's just minimizing energy, and that's the idea. But you should realize that when we represent this folding reaction, call it a folding reaction, it's no ordinary reaction. It's not like the usual chemical reaction where bonds are being broken or made. No covalent bonds are made or broken when you fold or unfold the population. What you're simply doing is dialing the population up or down in response to physical conditions by changing the energy. So as you put in more and more denaturant, you change the population from one fraction unfolded to a higher fraction unfolded, but you're not breaking any bonds. All you're doing is changing the population, and this remaining population, this tiny little population up here, still has the information in it that's necessary to encode the native fold. So I would argue that we should be making a distinction between stability and specificity, and that probably what we're doing in order to recognize the fold, although not necessarily to stabilize it under physiological conditions, is not trying to simply minimize energy. There's something else going on. Next thing that we all know, but I'm questioning whether we really do, is that the unfolded state is a featureless landscape. So this is based on work that was done by Paul Flory, theory that was done by Paul Flory, and then the great protein physical chemist Charles Tanford worked for more than a decade to find out the conditions under which a protein is really genuinely denatured, and showed by dint of many experiments that under 6-mole guanidinium, most proteins genuinely do obey random coil statistics. So let's go into this. Here's the model for the unfolded state. There are no strongly preferred backbone conformations. The energy differences among these different backbone conformations under unfolded conditions are very small, of order kT. So any given molecule in this population can readily visit any of these conformers. And of course, we all know that this isn't quite the right idea. It's a statistical coil, not really a random coil. But for purposes of this talk, random coil is close enough. And the way to think about this is like the diffusion equation, except with excluded volume imposed by the fact that you're dealing with a polypeptide chain, which takes up lots of volume. So it's kind of like Einstein-Smoluchowski 1905, but with excluded volume imposed. So what does this mean in practice? Here's a Ramachandran plot of a dipeptide. What you see is that just due to steric interactions between nearest neighbors, most of the space shown here as white is actually excluded because the electron clouds of two atoms in the dipeptide overlap in that conformation. It's very nice because for a dipeptide you can represent all the conformations in 2 dimensions. And what you see is that most of the population is focused here or there. And if you took a phi-psi angle from here and repeated it again and again, you'd get an alpha helix, again and again here, and you get a beta strand. Here's the polypeptide chain. Here are these 2 degrees of backbone freedom, phi and psi. And so in the style of Cyrus Leventhal, we can do a little calculation on how much conformational space would be available under unfolded conditions. Well, if each phi-psi pair can adopt only 2 values, being either there or there, and of course it can adopt more, then a chain of 100 residues has about 2 to the 100th conformations, possible conformations. 2 to the 100th is about 10 to the 30th accessible conformations. Remember, our dilute solution of proteins that we were dealing with in our reaction vessel might have about 10 to the 15th protein molecules in it. So 10 to the 15th protein molecules, 10 to the 30th available conformations. It's perfectly possible that no 2 proteins in that population have the same conformation at any given point in time. And yet when you switch to folding conditions, essentially all of them do. Well, so here is the reasoning. If each phi-psi pair can adopt 1 or 2 values, then a chain of 100 residues has 10 to the 30th accessible conformers. Where do we get off assuming that each of these residues is independent of all the other residues in order to do this sort of back-of-the-envelope calculation? And the place this comes from is the Flory isolated pair hypothesis, meaning that each phi-psi pair does what it does in isolation from all the other phi-psi pairs, with the exception of its immediate chain neighbors on either side. So this assumption that the phi-psi pairs are independent of one another lets you do the calculation. It makes the calculation very easy. But on the other hand, is it true? And Flory did an experiment which, if you go back and read what he actually said, he knew that this wasn't quite true, but he thought it was a pretty good approximation. So it's this simplifying assumption that each phi-psi pair is sterically independent of all the others. Now, when Rohit Pappu was a postdoc in the lab, he's now a professor at Washington University in St. Louis, but when he was a postdoc in the lab, he tested the Flory isolated pair hypothesis by an exhaustive calculation. What he found is that indeed it is quite true in the northwest quadrant of the phi-psi plot, but in fact it's not true in other regions of the phi-psi plot. In the northwest quadrant, when chains adopt that conformation, they're kind of stretched out in strand-like conformations, and it sort of makes sense that a residue here doesn't interact with a residue over there. In all other places in the phi-psi map, all other allowed places on the phi-psi map, the chain is much more contracted. And when it's contracted, it has opportunities for a residue to interact with other residues that are nearby, but not strictly nearest chain neighbors as Flory had thought. And that's what Rohit discovered. And so, having discovered that, it's clear that there's more organization in the main chain than we have previously thought. And the question is, what kind of organization? Nick Fitzke, when he was a graduate student, Nick has gone on to bigger and better things. He's now a postdoc with AdBax, and in fact, I see him sitting back there in the audience. When Nick was a graduate student in the lab, he decided to find out what kinds of interactions these might be. And he found a number of them. Here's a particularly dramatic one. An alpha helix can't be followed by a contiguous beta strand. What does that mean? It means that if you had 3 consecutive residues in this part of the phi-psi map, so here's alpha I minus 3, I minus 2, and I minus 1, and you take the next residue and put it here in that part of the map, you get a steric clash. So it's not a nearest neighbor steric clash, it's an I to I minus 3 steric clash, but nevertheless, it's a steric clash. Furthermore, it's a steric clash between the backbone carbonyl oxygen of this ith residue and the backbone carbonyl oxygen of the I minus 3 residue. So it's a backbone-backbone clash, which means that regardless of what residues you have there, since it's backbone-backbone, you will get that clash. Now this is an odd kind of result because it's very simple. So if you show this to a theoretician, the theoretician says, well, I know I could just relax the phi-psi angles ever so, and I could get rid of that clash. But you can't. And the demonstration that you can't is what Fitzke did. That's why this is a paper, not just a picture. So you can't get rid of the clash. And if you talk to an X-ray crystallographer who's spent a lot of time looking at proteins on the screen, they say, well, I think I knew that because I never see in a solved structure that a beta strand follows immediately after an alpha helix. There's always an intervening turn or loop separating them. And this is why. Why does this matter? Because these are the 2 most populated regions in the phi-psi map. And they're by far the 2 most populated regions in proteins. So it tells you that a huge number of conceivable conformers that you might have, if you could have either an alpha or beta, if you had that, many of those have to be eliminated because if you had 3 consecutive residues anywhere in this region of phi-psi space, the next one can't be there. So it imposes a kind of organization not just on folded proteins, but on unfolded proteins as well. And so it also shows in a very palpable way that there can be systematic local restrictions that extend beyond adjacent chain neighbors. Here's an I minus 4. Well, Dr. Fitzke went on to ask himself whether this is a singularity, and it turns out it's not a singularity. He identified many other kinds of conformations in which you either get a steric clash or alternatively, you get a conformation in which you couldn't solvate a backbone polar group, either by making an intramolecular hydrogen bond or a hydrogen bond with water. And that would be a high energy structure and would be eliminated, largely eliminated from the population, as we will come to in a moment. Well, if that's true, then what about all of this wonderful work that Flory and Charles Tanford did, showing that the unfolded, one can write an equation, a simple equation, here is that equation, for what the mean square radius of gyration would look like for a population in good solvent, if it's a random coil, and Tanford's subsequent work showing that in fact the folded structure satisfies this equation. So this equation says that the mean radius of gyration that you will see for this population goes as the number of residues in the polymer times some prefactor raised to an exponent, and a good solvent, that exponent should be 0.6. And in fact, that's what Tanford and subsequently other people like Sebdoniak and Kevin Plasko have found when they've done small angle X-ray scattering, where you can back this exponent directly out of an experiment. Nick Fitzke then said, well, I'll make up a contrived example and see how well this holds. So here's Fitzke's contrived example. You take proteins of known structure, an X-ray structure of a protein. You hold it exactly in its X-ray structure, except you allow it to wiggle in just a few places. Where do you let it wiggle? He identified it by an algorithm, but essentially you let it wiggle where the polypeptide chain turns are. So 92% of the protein is held exactly fixed in its rigid X-ray structure, 8% is allowed to wiggle. You now generate random chains by wiggling a protein of known structure at these wiggle points. When you do that, if you get a steric clash, you throw it away, and you keep doing it again and again until you get a representative population of random chains, but only randomized at this 8%. Oop. Only randomized at this 8% of the chain. Now, you can do a mean squared radius of gyration on that population. And you can do it for not just one protein, but you can do it for any number of proteins whose X-ray structures you know. And when you do, strangely enough, what you find, not so strangely really, what you find is that you get an exponent of 0.6. So these are certainly not proteins lacking organization. They are random coils in the strict sense of random coil. After all, when you think about it, anything is a random coil if you make it long enough. If I take a steel I-beam and stretch it from here to the moon, it's going to behave like a random coil. The question we're arguing about is at what chain length does it begin doing that? So what Fitzke found is that proteins that are held largely in their native conformation are nevertheless satisfy this Flory equation. So what you realize is a kind of broken syllogism. A hypothetical random coil that lacks any organization has a radius of gyration with an exponent of 0.6. Unfolded proteins also have a radius of gyration with an exponent of 0.6. And therefore, says the syllogism, unfolded proteins lack organization. But that's a broken syllogism. It's like saying, my first name is George and I grew up in Texas. George Bush's first name is George. He grew up in Texas, and therefore I'm George Bush. And it's broken, and I'm grateful for that. Okay. Next big question. Folding arises predominantly in the native side of this NTU equation. The side chains really have to do all the discrimination because the backbones are essentially all the same. So now we're going to take an excursion into adaptive biology. So the adaptive biology community has for years been studying organic osmolytes. And Wayne Bolin, Tom Record, several others have taken up the physical chemistry of these small molecules. So I want to talk about organic osmolytes for a moment. So what are organic osmolytes? They're small organic compounds. They have no net charge. They come in all kinds of delicious flavors. Here's trimethylamine oxide. Here's an amino acid. Here's a sugar. Here's another sugar. There are alcohols. Here's urea, the common denaturant. It's an organic osmolyte as well. These are found in all species in nature. They're found in all three kingdoms in nature, and they were there from the very beginning. And what do? What they do, the adaptive biology community is interested in them because they provide some kind of organic ballast to keep the cell from rupturing osmotically. But what they do to proteins is they can fold and unfold proteins. These are protecting osmolytes. They fold proteins. That's a denaturing osmolyte. It unfolds proteins. There's water put in there as a kind of fiducial marker. All kingdoms have these osmolytes. If I go out into the desert and find a plant that exists in the form of one of these desiccated seeds that's been living there for 100 years under hot desert conditions, and you add water to it, and the plant blooms, why don't the proteins in the seed denature under those incredibly denaturing conditions? And the answer is because they have organic osmolytes, protecting osmolytes that are packaged up in the seed that do that for the plant. When I don't drink for a while, the concentration of urea, this denaturing osmolyte, the concentration of urea in my kidney can go up to 2 or 3 molar. In a shark or a ray, it can go up to 5 molar. 5 molar urea is enough to denature most proteins. Why don't the proteins in our kidney denature? And the reason why is because they're accompanied by protecting osmolytes. In the shark, it's accompanied by TMAO, which is poisonous for us, but we have glycine betaine. And those are counteracting protecting osmolytes that protect our proteins from denaturation as a consequence of urea. So there's a lot of work that's been done on the thermodynamics of osmolytes, and what we know is that protecting osmolytes shift this folding equilibrium towards the end state, and denaturing osmolytes shift the equilibrium towards the unfolded state. This one is protecting, these are all protecting, that's denaturing. And here's a review article with Wayne on this whole topic. But most of the work in this area has not been done by me, it's been done by Wayne or Tom Record or others. And I'll show you some references. How is it, thermodynamically speaking, that these osmolytes are doing this? And what they know, and we're confident that we know this because it's straight thermodynamics, it's been measured. And it's been measured by a number of investigators. What we know thermodynamically, not mechanistically, is that the way they're doing it is that if you had a population of proteins under denaturing conditions, so here the unfolded state in water is lower in free energy than the native state, and you put in a protecting osmolyte, it raises the free energy of the native state, but it raises the free energy of the unfolded state much more, so that now the native state is lower in free energy than the unfolded state, and it folds the protein. We're saying nothing about mechanism. And exactly the converse is true for a denaturing osmolyte. It lowers the free energy of the native state, but it lowers the free energy of the unfolded state a whole lot more, so now the denaturing osmolyte can change a population in which the protein is the folded population dominates to one in which the unfolded population dominates. Again, thermodynamically, what we know is that the predominant effect is on the unfolded state, and it's exerted predominantly on the backbone. It's not on the folded state, and it's not on the side chains. So whatever osmolytes are doing, and they're doing it, and we know this thermodynamically, they're doing it to the backbone, not the side chains, and they're doing it on the unfolded state, not the folded state. And if they're doing it to the backbone, it makes perfect sense. It's plausible that if what they're doing is predominantly to the backbone, it should be also predominantly on the unfolded state, because in the unfolded state there's more backbone exposed to solvent than in the folded state. This work was done initially by Serge Timoshev, and then more recently, here's a paper by Matt Outen and Wayne Bolin. I should emphasize that in coming to these conclusions, which are sound thermodynamics, there were many, many complex problems that had to be solved along the way. And I don't have time to go into this, but doing this in a way that is thermodynamically sound turned out to be a real tour de force. So the next thing to realize, here are a number of references, is that the effect is universal. That is, proteins don't have binding sites in their backbones or anywhere else for this broad repertoire of osmolytes. And yet they all do this. Some are better at it than others. These are arranged in order of the effect. So TMAO is a much better protecting osmolyte than, say, betaine, but nevertheless, they all operate in the same way. Proteins have no binding sites, no specific binding sites for these. So whatever is going on, it's some kind of universal effect. So all of this is thermodynamics. It then became irresistible to Tim O'Street, who was then a graduate student in my lab. He's now a postdoc with David Agard at UCSF. It became irresistible to Tim O'Street to ask why. And he made up a statistical thermodynamic model with 27 microstates that seems to account for the details. It's a rather simple model. And the bottom line from this is that molecular origin of the effect really has to do with peptide hydrogen bonding. And that makes sense. The predominant effect is on the backbone. The backbone is polar. Every residue has a carbonyl oxygen and an amide hydrogen that need hydrogen bond partners. What you see from these osmolytes is that urea has a very polar surface, but these protecting osmolytes have some apolar surface and some polar surface. If you just look at the fraction of apolar surface, the fraction of apolar surface goes as the strength of the protecting osmolyte. And what happens is sometimes, in the presence of osmolyte, the osmolyte is turning its apolar surface to the backbone, keeping the backbone, inhibiting the backbone from making a hydrogen bond with water, and therefore it has to make a hydrogen bond with itself. And so it pushes it towards the folded state. What this is really doing is the osmolytes are making water a better solvent or a worse solvent than pure water. So if water is a really good solvent for a polypeptide chain, the chain kind of reaches out to the solvent and says, take me, solvent, whereas in a really poor solvent, it shrinks back and interacts preferentially with itself. And these osmolytes are modulating that effect. That is, in fact, what we think, even if we're wrong. So that's a model. But the rest of this is model independent. It really depends upon pure bulk thermodynamics. So the reason why this is important is if what you thought is that the backbone doesn't differentiate anything and that most of the folding takes place in the end side of the equation, this says just the opposite. Osmolytes may work differently than protein folders, but what osmolytes do in all three kingdoms of life and in every species is they fold proteins by interacting with the backbone in the unfolded state. Well, how about the hydrophobic effect? We know that the hydrophobic effect is the driving force for folding the protein. Every textbook tells us that. How do we know? Here's the reasoning, which makes perfect sense. Here you have a polar group in the unfolded state. Most of the backbone polar groups in the polypeptide chain are satisfied by making hydrogen bonds with water. In the folded state, they're making hydrogen bonds, some of them with water, some now intermolecular hydrogen bonds. But a hydrogen bond is basically a hydrogen bond, whether it's with the protein or whether it's with water. So this is kind of a wash. On the other hand, in the unfolded state, you have a lot of exposed apolar groups. Whereas when you fold the protein, these hydrophobic groups can cozy up to one another in the core of the protein. In fact, we believe they engender the core of the protein. And now what happens is they're removed from solvent access, and we know from work that dates back a long, long time and was put on a really strong footing by Tanford and by Wolfenden, that in fact the free energy of transfer of these groups from water to an organic medium is very favorable. And so we can really quantify this, and Walter Kautzmann in his famous 1959 Advances in Protein Chemistry article showed us that this had to be a very, very strong driving force. But one of the things to recognize is that, so nobody argues that it's not a strong driving force, but one of the things to recognize is that in folding the protein, there's only one of these big three forces that kind of pivots between the folded and the unfolded state. Conformational entropy always favors the unfolded state. You lose conformational entropy when you fold the protein. That's unfavorable. The hydrophobic effect always favors the native state. Shielding hydrophobics from solvent access is always a good thing. At higher temperature, the hydrophobic effect becomes even stronger. So it's really only hydrogen bonding that favors the native state under folding conditions and favors the unfolded state under unfolding conditions. And recognize that osmolytes shift the unfolded population by operating on the polar backbone, which we believe means operating on the hydrogen bonding that's going on in the polar backbone, by either making it more favorable for the protein to hydrogen bond with itself, or more favorable for the protein to hydrogen bond with its aqueous surround. So hydrogen bonds are definitely a major organizer of conformation. And the next thing you have to realize is that all folded proteins are built on hydrogen bonded scaffolds of alpha helix and beta sheet. There are some small exceptions to be sure. Some of these small, very small metal binding weirdos don't have alpha helix and beta sheet scaffolds, but everything else is built on one of these scaffolds. And it must be. You realize it must be. Because what happens is that if you take hydrophobics out of water and bring them together on the inside of a protein, so you create an inside and an outside for the protein, then inescapably the hydrophobic side chain is attached to a polar backbone, and the polar backbone gets transported to the inside. And if that polar backbone could make hydrogen bonds with water when it's unfolded, but couldn't make hydrogen bonds with anything when it's folded, then that would be a broken hydrogen bond. And as I'll try to show you in a moment, that would be so costly that you wouldn't fold the protein. And as Ramachandran showed in 1968, there are really only two extensible ways of making hydrogen bonds for the backbone. You can put them in alpha helix, or you can put them in strands of beta sheet. And any other way that you try to extend the chain turns out to lead to a place where you're going to have a defect with unsatisfied hydrogen bonds. So, they're built on scaffolds of alpha helix and beta sheet, and it's no accident. Now, can you afford to have unsatisfied hydrogen bonds? And I'm going to argue that you really can't. So Pat Fleming, who was a research associate in the lab for a while, he's now a research professor still at Hopkins, examined this issue. And the idea is this. There's an argument in the field. In fact, it's a rather strange argument given that we're in the 21st century. People still argue about the effect of hydrogen bonding in proteins. There's one group that argues, based on calorimetric arguments, that a hydrogen bond, an intramolecular hydrogen bond, stabilizes a protein relative to making the hydrogen bond with water. How much? By about a kcal. Another group argues that no, in fact, just the opposite is true. Making a hydrogen bond in a protein actually destabilizes the protein relative to making that hydrogen bond with water by about a kcal. And so they argue back and forth. No, it's a positive kcal. No, it's a negative kcal. And that argument, which has predominated in the literature, misses a really important point, which is that if you could make a hydrogen bond with water in the unfolded state, but couldn't make a hydrogen bond at all when you folded the protein, that's not 1 kcal positive versus 1 kcal negative. That's a broken hydrogen bond, like a broken hydrogen bond in the gas phase. And so from quantum mechanics, we would believe that that is of order 5 kcals, 6 kcals. Nick Pace tells me that that estimate is probably too high. It's probably only about 3 kcals. But in any case, there's a substantial cost to an unsatisfied hydrogen bond. If it's 5 kcals, we would expect that the population of unsatisfied hydrogen bonds in proteins is of order about 0.02%. If we actually analyze proteins and ask what fraction of the backbone has unsatisfied hydrogen bonds in proteins of known structure, it turns out to be 10%, not 0.0002. So what's going on? The analysis seems pretty straightforward. It seems unlikely to be all that wrong. So how can this be? So we've looked at proteins with this eye towards asking, how can there be so many unsatisfied hydrogen bonds in proteins? And the answer to this is that in many cases, the crystal structure is just wrong, but for understandable reasons. So you have an amide in the crystal structure, and here's the nitrogen side chain, nitrogen part of the side chain, and here's the oxygen. And if you just flip them, they would now be making hydrogen bonds, whereas they weren't before. So that's a really trivial case. But there are other more complicated cases where what you're seeing is that the criteria you're using to assess a hydrogen bond, even the most liberal criteria, which are, after all, geometric criteria, don't recognize certain conformations which really look like they should be hydrogen bonded, but they are outside the hydrogen bond constraints imposed by anybody's function. So here's an example. This is work that was done by Nick Panasic, When he was a postdoc in the lab, he's now a professor at Claflin College in South Carolina. And Nick Panasic recognized that if you looked at the coil library, what's the coil library? If you throw away all the alpha helix and all the beta sheet, what's left is the coil library. And Nick Fitzke systematized this, and it actually is a very nice utility, and it's available on the web if you want to look at it. No matter how you do it, you throw away all the alpha helix, all the beta sheet, and what you're left with in the protein folding community is called coil, which is crummy nomenclature, but for historical reasons, we're stuck with it. I mean, it's obviously not coil in any sense that a polymer chemist means coil. And you can see it in the X-ray structure, so it's not random. Nevertheless, we call it coil. So when you analyze this, you find that about 25% of the coil library, maybe a little more, is turns. And there are then many conformations that look very close to turns, but no matter whose hydrogen bonding criteria you use, they're a little bit outside the range of what you would expect for a proper turn. So we call those turn wannabes. And the question is, if we had taken the turn wannabes and done a little tweaking to the turn wannabe, could we tweak it into a good hydrogen bonded turn conformation? And the answer to it is yes. So what Panasic did was he excised this segment from the protein of known structure. He fixed the ends, a little bit N-terminal to where the turn is, a little bit C-terminal to where the turn is. He fixed the ends. He did local torsional angle minimization on the intervening segment and minimized into good hydrogen bonded turn geometry. And what he found is, here's a worst case. You change the locations of the atoms. You displace the atoms by at most about an angstrom, and frequently much less than that. If you now go back to the X-ray structure for which you had structure factors and ask, did you move out of electron density when you did that? The answer is no. You understand why this is true. When you solve an X-ray structure or an NMR structure, you have no explicit hydrogen bonding potential in that structure, and for very good reasons you don't. But nevertheless, there are these conformations which we expect are really forming hydrogen bonded turns, but that's not what you find when you look at the PDB. If you minimize all these and take the ones that you really believe in, you increase the apparent population of turns by about 13%. And the whole point is that this idea that maybe 10% of the backbone hydrogen bonds are unsatisfied in proteins, we suspect that that's simply not true. And here's a case study which tends to demonstrate that. So we think that hydrogen bonds play a really significant role in all of this as an organizing force, and in more than one way, which I'll get to in a moment. So now, Lauren Persky, a graduate student in the lab. I will no longer be able to say graduate student in the lab after tomorrow, because tomorrow she defends her PhD. So after tomorrow, it will be Dr. Persky. So here's what Dr. Apparent Persky did. She wanted to look at the phi-psi angles, the distribution of phi-psi angles in the coil library, no helix, no sheet, which have lots of hydrogen bonds in them, and assess how much hydrogen bonding is actually going on here. So if you plot the phi-psi angles that you see from the coil library, you get a distribution that looks something like this. So what do you make of this? You don't make very much of it, but if you contour this, what you see is that this population falls into basins, very clear basins, some of them with real bullseyes in them. And if you draw little outlines around the basins, there are 7 basins, and it turns out that every one of those basins is associated with 1 or maybe 2 local hydrogen bonded motifs, with the sole exception of polyproline 2. So hydrogen bonds are clearly playing a strong organizing role, not just in alpha helix or beta sheet, but in the coil library as well. And Lauren then went on to ask the question, well, what are the physical chemical determinants of these basins? And what she did was to then analyze the basins using simple Monte Carlo simulations. And what she found is that if you select the very small segments from the coil library, 2 and 3 and 4 residue segments, and look at the fraction of population covered by the top 40 conformers, that in fact there are 23,000 segments. Of those segments, there are 1,500 unique conformers among them. But 72 of those conformers account for about 2/3 of the whole population you're seeing. And one of the reasons why this kind of analysis has been complicated in the past has been because we include all of the conformers, but many of them are very rare. Here in this population, what we see is that approximately, well, more than 600 of these conformers occur only once in the whole PDB. Is that a mistake, or is it really important, even though it occurs just once? I don't want to argue that, but what it does mean is that it doesn't account for very much of the population. And so what Lauren did was to, I think quite sensibly, throw away this population of very rare conformers, and anything that appears less than some small fraction, than less than 0.05% in the whole PDB is thrown away, and she deals with this population of conformers. And you can see the top conformer accounts for more than 25% of this whole library. The next conformer accounts for about 18%. So the top conformers, the top 5 conformers, account for most of what you're seeing. And you can go all the way down to approximately 35 conformers or so before you get into this very rare population. And what she did was to do Monte Carlo simulation and ask how many of these conformations could be reproduced by very simple criteria. Sterics, meaning you can't allow 2 residues to occupy the same place at the same time. Some kind of hydrogen bond score, which is really a score and not a potential. And importantly, this requirement, which gave rise to the title of this talk, which is that all backbone polar groups, for reasons we just went through, have to be hydrogen bond satisfied. Because an unsatisfied hydrogen bonding group in a little segment like this or in a whole protein would cost you about 5 kcals, and that's equivalent to the whole free energy difference between the folded and the unfolded state. So you can't afford very many of those. And what you find is that many of these conformations are eliminated because they shield the backbone polar group from solvent access. So she found that 98% of these segments could, after throwing away the very rare ones, could be reproduced by just these 3 very simple criteria. No hydrophobic effect in any of it. And that furthermore, 90% of the longer coil segments, all the way up to 20 residues or so, could be covered by composites of these shorter primitives. So here is an attempt to come up with real physical chemical understanding of these short segments. If we really had that kind of understanding, then instead of taking a fragment library out of proteins of known structure, we could simulate a fragment library. So this is the real organizing power of unseed interactions. All of the conformations that you could imagine that aren't built on hydrogen bonded scaffolds of alpha helix or beta sheet would be eliminated in proteins for reasons we just went through. These small coil conformations, which are not alpha helix or beta sheet, many of them are also eliminated just because they would be hydrogen bonded satisfied. So that's not because of an enthalpically favorable interaction that you could see in the folded structure. It's because of an interaction that doesn't occur. So you never see it because it didn't occur. But if that is so unfavorable, it would make a negligible contribution to the overall thermodynamic population, and you wouldn't see it in the final state. So I want to conclude by just telling you a point about a whole parallel universe. So everything I've told you about so far is based either on analysis of X-ray structures or computer simulations that were done in our lab. A year and a half ago at a Gordon conference with Buzz Baldwin and Carl Frieden, we got to talking about all this and about the whole experimental track that exists. And we decided to put it all together, all the experimental literature together, and come up with an understanding of what must be going on. So a very diverse set of experimental findings was wedded together in a paper in which we talked about dry molten globules. What's a dry molten globule? So a dry molten globule is a structure in which it's an expanded form of the folded protein. The overall chain topology is there, the hydrogen bonded secondary structure is there, the solvent has been squeezed out of the, or at least mostly squeezed, out of the interior of the protein, which is why it's dry. Therefore, the hydrophobic shielding is already developed, but the side chains haven't yet locked. So what that means is that all of these characteristics of the native fold are there without the close packing of side chains that we all thought were the things that selected the native fold from the rest of conceivable fold space. So those characteristics are all there. Am I just making this up? Is this a real state? What this says is that you don't need that state in order to get this. Instead, you would have a fluffier, more expanded state in which you could also get these characteristics. And so we decided to put all this together in a paper. There's the paper, I say we. This is 90% Buzz Baldwin and 10% Carl and George. Buzz, for those of you who know him, has this wonderful ability and an essentially photographic memory to synthesize these experiments that come from so many disparate places. And what we showed in the paper is that there are 4 examples, one from Buzz in 1995, one from Carl in 1995, and 2 more recent ones, one from Jayanta Dankar in 2009, one from Thomas Kiefaber in 2010, and all of these show clear evidence of molten globules. Why hasn't this been seen previously? Because the standard tests for the folded state of the protein, like trip fluorescence, wouldn't distinguish between a molten globule and an unfolded protein. The molten globule, after all, is dry on the inside. So the quenching of a trip doesn't change between the molten globule and the native state. So in fact, the usual probes are blind to differences between the molten globule and the native state. But if this is true, and there are a number of tests that are currently underway, then the 2-state model that we've all been using breaks for those proteins that fold or unfold by way of molten globule intermediates, or so either fold or unfold by way of molten globule intermediates. And in fact, our hypothesis is that many more proteins besides these 4 do exactly that. It's an experimentally testable proposition, and tests are underway. So keep your eyes peeled. We're interested to know whether this is going to happen or not. We think the evidence is pretty strong. So summarizing the conventional model, unfolded state is a featureless landscape. Folding arises at N, and all backbones are the same. The side chains discriminate. Our model instead is that the unfolded state really is far from featureless. There's a lot of organization in the unfolded state. People are actually beginning to find this now that they look. What we know from osmolytes is a lot of organization arises in the unfolded state, and most of the organization arises not because of what goes on in the side chains, but because of what goes on in the backbone. There are only a small number, like a few thousand possible backbone structures that are available for a single domain protein. And then what the side chains do is discriminate, but amongst that set of possibilities. But that set of possibilities is winnowed down by hydrogen bonding in the backbone. So essentially, this stands the current model on its head. And there's a paper that describes some of this. It's a perspective in PNAS, a controversial perspective, I should say. And with that, I should remind you of the people who were responsible for this work. I mentioned their names as we went through it. Lauren, soon to be Dr. Persky, is just finishing up in the lab, did this work on the physical chemical determinants of short segments. Pat Fleming was responsible for looking at the contributions of hydrogen bonding, both thermodynamically and in proteins. Nick Fitzke did all of this nice work on the unfolded state. Nick Panasic looked at whether there's a larger population of hydrogen bonded turns after a little minimization. Tim O'Street made up this nice thermodynamic model, this nice statistical thermodynamic model for what osmolytes are doing. I didn't mention Teresa's work. Teresa's work has really set the stage for our understanding of the conceivable number of protein domains by work that she did when she was a postdoc in the lab. Rohit did the initial work on the unfolded state. He's now a professor at Washington University. And Raj figured into a lot of this. He went back to India some years ago. He's now the head of a big software group at Tata Consultancy. And my collaborators, Buzz Baldwin, Wayne Bolin, and Carl Frieden. Thank you all for staying light. Do you do questions after these things? I could turn off the mic and we could do questions if you want.
Just before we put this up here, there's a reception on the right-hand side, and here's the question.
So that was a terrific talk, but I'm a little bit, in my mind's eye, I'm having trouble envisioning the status of side chain hydrogen bonds in the molten globule. If there's such a penalty for losing backbone hydrogen bonds, what happens, how do you minimize the loss of energy for putting the side chains into the interior?
So if you analyze side chain hydrogen bonds in proteins, it turns out that most of the side chain, the hydrogen bonds polar side chains make, are with backbone. Many more with backbone than with side chain hydrogen bonds. And most of them are really quite local. So what you find is that a side chain is hydrogen bonded to either its own backbone or the backbone of a residue two or three residues away. You see this very strongly in helix capping at the beginning and ends of helices, but you see it in other places as well. And so what are the side chains doing? I think that as the secondary structure has formed, including the structure that comes from the coil segments, the side chains have already satisfied most of their hydrogen bonding requirements by hydrogen bonding with backbone elements that are already in place. In fact, if you look at that salt bridges between side chains, the number of actual salt bridges, even though there are many charged polar residues in proteins, they're typically sticking out in water. And the number of actual salt bridges in proteins is fewer than one per protein. So these things are not as common as you might think. You can introduce them, but in naturally formed proteins, they're really quite scarce. Alan.
Going back to the Ginsberg and Carroll paper that you mentioned early on, would you not agree that if you have many different properties, independently measured properties, that follow the same fractional change on exposure to unfolding conditions, would that not be evidence of a true two-state thermodynamic, at least in the thermodynamic sense, a true two-state transition?
Well, so what you're looking at is a two-state transition from the unfolded state to something. But since what you're using are spectroscopic probes that are not distinguishing between a dry molten globule and a native protein, that something might well have been a two-state transition from the unfolded state to the dry molten globule. Why is that important? Why is that? Are we just talking about the number of angels that can balance on the head of a pin? And the answer is no, because if it's true, it's a profound realization that you can get all of these organizational characteristics for the protein kicking in without having the side chains actually lock. So that actually, although it seems like it's making the problem more complicated, is really simplifying in terms of mechanism.
So you're saying that experimentally, how do you distinguish experimentally between a dry molten globule and a true native state? Enzyme activity?
Well, I would suspect that enzyme activity might do it, and I know you've used enzyme activity, but typically people aren't as refined as you are. So Here's the way Jayanta Dhankar did it, for example, in 2009. He used a FRET probe. So what he did was he starts to unfold his protein in guanidinium in this case. Too bad he didn't use urea. And what he attaches FRET probes at different places in the molecule. So he could see that, for example, a helix in the molecule is moving as a rigid unit with respect to other places, but he could also see that the protein is still dry because ANS doesn't come to the interior. And the other thing he does is introduce FRET probes at various distances in the protein, and he could see that they're moving apart.
So in other words, you wouldn't trust any spectroscopic, ordinary spectroscopic or hydrodynamic technique for distinguishing between the native and a dry molten globule?
You would certainly not trust something like trip fluorescence. If all the CDs, if the hydrogen bonded backbone structure is in place, then CD won't distinguish. A hydrodynamic method, if you're looking at the hydrodynamic radius or using small angle X-ray scattering or something like that, you'll see a little bit of expansion maybe, but it's typically pretty blurry. But the one method that seems to be doing it is something that Kathy Royer is doing with pressure denaturation of proteins. And there she could see very clearly as you begin to pressure denature the protein that it goes to a slightly puffed up state, and in that slightly puffed up state you could begin to make a number of measurements. So that one is more direct, but most protein biochemists don't have, rightly or wrongly, the same kind of intuitive feeling for pressure denaturation that they do for chemical denaturation. I should say that Kathy Royer points out that they probably really don't know any more about chemical denaturation than they do about pressure denaturation either, but familiarity breeds confidence. So.
But pressure denaturation reflects an actual volume change of the entire system, as you point out. So it involves not the actual, it involves a change in exclusion of solvent by the protein as the protein unfolds. Is that correct?
Yes, but
some differential exclusion.
Differential exclusion, but the other thing that you could see is that the interior remains dry. So.
Yeah, so what do you think the activation energy barrier between the native state and the dry molten globule state is to really qualify it as a real thermodynamic state?
Right, so that's a great question, and I don't know the answer, but let me expand on it just slightly so that everybody else will understand what you just asked in case they don't know. So people think that there's a transition state in a protein. It folds from the unfolded state to the folded state. There's some kind of barrier there. And people have represented that transition state the way you would represent a transition state from absolute rate theory. That's probably not correct. There are no covalent bonds being made or broken. But nevertheless, there does seem to be a barrier there because the difference in free energy between the folded and unfolded form is 5 to 10 kcals. If you distributed 5 to 10 kcals across 100 residue protein and you distributed it homogeneously, then no residue is stabilized by anything larger than kT, and consequently the protein would just unravel. So there must be some barrier somewhere. The evidence seems pretty strong that that barrier is not between the molten globule, the dry molten globule, and the native state. It probably occurs before that. But precisely what it is, we could speculate, and I do, as you know. That's Nick Fitzke, by the way, for those of you who don't recognize him, who did all this really nice work on the unfolded state and is now doing much nicer work at Adbax's lab. So we could speculate, but I don't know the answer to that in ways that are as crisp as the way I've just tried to describe these results today. But the one thing we think we know is that whatever it is, it occurs between the unfolded state and the dry molten globule. And there's evidence for that.